Hello, and thank you for joining us. Today's webinar topic is Maximize Workflow, Automating High-Volume Protein Purification for Medium Pressure Chromatography Instrumentation. Our presenter today is Pete Winship, Regional Sales Manager for Teledyne Labs. If you have any questions, please feel free to submit them throughout the presentation, and Pete will respond after the webinar concludes. This webinar is being recorded and will be available within a few days. Okay, now I'm going to turn it over to our presenter, Pete Winship.
Thank you for the kind introduction. Hello, everybody, and thank you for joining today. My name is Pete Winship. I'm one of Teledyne's regional sales managers. I cover Europe, the Middle East, and Africa for certain products for the Teledyne Labs division. So, which I'll go into in a moment. My background, so that you know, I come from an analytical chemistry background. So this is my area essentially. What I'll be bringing to this is certain aspects of analysis and automation that I've been involved in over several years now. So, and as mentioned today, I'll be describing automating workflows for protein purification, where we're using medium pressure chromatography instruments to meet that end. So I mentioned Teledyne Labs, so just a brief moment on who we are. Teledyne is a very large corporation, and Teledyne Labs is one of the divisions of that corporation. And essentially we are 5 companies, so here CTAC, Tecmar, ISCO, Hansen and Lehman Labs. And these companies are associated with different aspects of analysis, instrumentation, automation, components, etc. Today I'm going to be focused essentially on automation, but particularly with the Teledyne CTAC Labs, Teledyne CTAC Technologies aspect of Teledyne Labs. So just a brief bit on who CTAC is. This is a company, so the CTAC brand that was founded in the mid-80s in the US. Essentially that started with development and manufacturing of an ultrasonic nebulizer system for trace element analysis work. It moved on to developing and manufacturing automation in the early 90s. And then the company changed hands a number of times before it was acquired by Teledyne Technologies in 2013, so 12 years ago now. So today, through Teledyne Labs manufactures automation, sample handling, sample introduction systems under this brand, and they do this for a range of different analytical technologies and workflows. And they do this in Omaha, Nebraska in the USA. So a quick agenda for you. I'm going to make a couple of points on using automation for analytical workflows. And then I'm going to move on into protein purification, the separation and isolation, etc. of high quality proteins, why we do this, how we do this, and then we're looking at an aspect of chromatography for this, so protein preparative purification, and then how we can maximize efficiency with automation for these workflows. So automation and analytical workflows. Just a brief note on this. I mentioned at the outset that my background is in analytical chemistry. One of the things that you would commonly expect in development of instrumentation for analytical techniques, etc., is that sort of from the early days of techniques, instrumentation, through to sort of their development, and as they get older, they get new generations of these techniques. You would typically expect a form of automation to come into that. Some techniques have gone from being essentially somebody introduces a sample into a system in the early days, and then over the years and decades, they might become entirely automated. So there's many different flavors essentially of automation for analytical workflows in a laboratory setting. Automation could look like something as simple as moving a sample around. It could be providing something like temperature control. It could be agitating the sample before it's used. A form of preparation, so that could be diluting a sample. It could be spiking a sample with a standard or similar. It could involve collecting something, an aspect of the sample, so fraction collection from a separation, for example, like in chromatography, or it could involve a form of introduction. That automation might be part of the methodology, so it might be a necessity, certainly in today's laboratory, or it might be some aspect of improving a workflow. So it might be looking to improve sample throughput, so more samples through your instrument in a unit time. It might be about adding a capability, or it might be, certainly today, it might be about reducing labor, effort, and cost and time saving. So certainly something which has been recognized post-COVID is that automation is becoming more important in laboratories and getting more effective use of laboratory staff time, putting their skill set into other aspects of the day-to-day operation in a lab. So for today, essentially where I'm going to focus is excuse me, sample introduction, and then how that sample introduction can support sample throughput in the laboratory setting, and then labor costs, time saving, etc. So that's where we're going to focus today. So isolation or separation, purification of protein species or large volume species. The why do we do this? Why is this of interest to us? So essentially, I'm sort of, you'll have to forgive me, I'm going to use the term protein or large molecule a little bit interchangeably here. Essentially, what we're looking at here is large molecule or protein demand and biotherapeutics. So this is something that was once considered a small aspect of medical treatment, and over the years and the decades that have followed, this has grown from being, like I said, that minor aspect into something absolutely immense. A global demand for therapeutics that have a biological origin, that are used in some form of therapy. Now, we will all know, it might be a family member or a friend or somebody we've known over the years, somebody who has had a need for a biotherapeutic, and it might be something like a therapeutic protein, a monoclonal antibody, recombinant protein, etc., etc. We've all known people who've needed something like this, and as people live longer, healthier lives, the diseases that people are treated for, certainly when they are older, this is where biotherapeutics come into this. Now, the trajectory of demand for this, essentially we're sort of on a doubling trajectory of demand for these kinds of species over a 10-year period. So in the lead up to COVID, the COVID era, essentially we were looking at a very large growth in demand for these species, these molecules, these protein treatments, etc. We were looking at essentially a doubling trajectory, and that has gone through COVID. That certainly has not gone away. And if you look, there's various sources of information on this, but if you started at, say, 2018, then you'd be looking at that doubling trajectory through to 2028. If you started today, then that doubling trajectory would be expected to see the same doubling through to 2035, for example. But basically a growing demand for these species. Now, what are we talking about? So some of these will be, if this is not your area, these will still be familiar to you. So some examples of the kind of species, the large molecules that are needed for therapies. Factor VIII, for example, this is used for people suffering haemophilia. Streptokinase, this is a species for the breakdown of blood clots. I've certainly got a family member who's had this in the past. Alpha interferon, this is used in certain types of cancer treatment, leukemia being a good example of this. Human growth hormone, insulin, this is another large demand. I'm sure that most people will know somebody who has diabetes, and this is something that's needed for that treatment, etc., etc. There are so many species, so many molecules, proteins, etc., that are needed for these therapies. So how do we isolate these things? Where do these species, these proteins that we're interested in, where do they come from? Now, there's many ways of doing this, many ways of isolating any kind of species, but one of the common ways to do this is via a form of chromatography. So separation with some form of column and instrumentation with a mobile phase that's working with this column to separate out the species of interest. So the protein purification workflow, essentially what you're looking at here is, in a nutshell, you're looking to isolate a species of interest which has its origins in some form of from a biological expression platform. So this might be a cell line, which has been cultured, and from these cells, you're looking to isolate and purify species of interest. So, and typically you need a lot of this. So you need to take your cell line, you culture it, you isolate what you're interested in, purify large amounts, and then you use that in either in research and development to further what you're doing, or you're using it in some form of end application. Now, there are various workflows, various systems, instruments, columns, conditions, specifications, etc., that are used to do this, but they all share a commonality as you work through it. And we can automate aspects of this. So what we can do here is we can essentially split this kind of work, this kind of separation, into two approaches. It's sort of a bit rough and ready, but we've got preparative purification. This is the one that we're interested in, which I'll speak to today. So you're looking at larger volumes, and then you've got analytical purification where you're working with smaller volumes. Now, if you sort of roughly separate these out in terms of volumes for preparative purification in the large volumes, you're looking at anything from, say, a couple of milliliters upwards, up towards, let's say, a litre. And then for your analytical purification, you're looking at small volumes of probably less than a milliliter. So that's your sort of threshold of split between the two. Today, I'm going to focus on the preparative because that's where our automation will come into the kind of volumes that we're going to work with. So instrumentation for preparative purification, there are several types of instrumentation that you can use for this kind of work. Two of the most common that you will see in laboratories around the world are the ACTA instrument from Cytiva or the NGC instrument from Biorad, and there are various versions of these instruments. They use various different types of columns and specifications and conditions, and they all support different types of workflows depending on the kind of project that the laboratory is working on at that particular time. Now, again, in a nutshell, what is the workflow for isolating what you're interested in? So, as I mentioned, you start off with some sort of expression platform. So you take your culture process, and essentially what you need from this culture process is you need some samples. So you're going to culture what you're interested in, and then at some point you're going to transfer your samples from plates or flasks, and then you're going to move them to tubes or bottles, which can then be used for the introduction process. Now, you might be interested in, at this point, single samples or a handful of samples where you're introducing them manually, or you're putting them onto some sort of loop system, multiple loop system, so you can get a handful of samples through sequentially. Your instrument then does a separation or purification process, so by one of these instruments, for example, or another. Once they've done the separation, your system will then essentially, you'll be looking to collect an aspect of the sample that's gone through. So the fraction that you're interested in that will contain the protein species or large molecule species of interest. And that's typically another form of automation as well. Now that separation process, this takes time. And the larger the sample volume that you're putting through, the longer the amount of time it takes to do your separation purification process, arriving at the fraction that you're interested in. So time, this is one of the aspects of working in a laboratory that if it can be put to better use, more efficient use, certainly in terms of your staff, this is very beneficial to workflows. So how do we maximize the efficiency of this process? So this is where we can come in for this. So this is the workflow that I've just described. Essentially, we start off on the left-hand side of this screen here. We start off with culturing, sample preparation, and then we move over to the instruments and we introduce our sample. The instrumentation does the separations that we're interested in, and then we capture the fraction we're interested in studying further. So simple. Now, as I mentioned, that would be a situation where somebody in the laboratory is doing an individual injection, or they're loading up some loops so that they can leave several samples waiting essentially in a queue to do the introduction to the system. So how can we improve this process? So essentially we can put our automation in the middle of this. So once the culturing aspect of this is complete and samples have been transferred to, say, sample tubes or bottles, which are shown here on the left, those tubes or bottles can be put onto our automation, and then our automation is connected to the instrumentation, and that can do all of the introduction aspect of this. So instead of having individual samples or a handful of samples, we can be talking about racks of samples which can then go through the system. So we go through essentially the same process, but the automation takes the place of the person that's doing the introduction aspect. The instrumentation still does the separations and isolations that you were expecting before, and we still collect the fractions that we're interested in. So essentially this takes the place of that person in the laboratory. So as I mentioned earlier on in this webinar, this is a global demand. So all over the world there are laboratories working on this kind of isolation and purification of species of interest. Now these, just to give you an idea, these are two photos, and oddly they look very they look very similar. Actually, it's the same kind of workflow here, but these are two laboratories on opposite sides of the world. Both I've visited. The UK, I won't identify these, but the laboratory on the left is one in the UK. I've visited several times over the last few years, and they are automating their introduction to an ACTA instrument for their isolations. The one on the right, very similar kind of laboratory. This one was actually in Australia, and I visited this one in 2022 as lockdown was being lifted from COVID. Essentially, they're using the same kind of automation. These are two different types of autosample, which I'll explain, but they're using them for the same reason, for the introduction of samples to improve their workflow and their efficiencies. So what we've got next is we've got some footage of the actual operation of this process, so automating the introduction phase to a chromatography instrument for protein purification. In this case, the example here is one of the ACTA instruments, but I'll I'll just walk you through what we've got here. I'm not going to be able to talk fast enough to go through this this footage, so I'm just going to sort of start and stop this in terms of the actual footage. So what we've got here is you've got, as you can see, the instrumentation, the chromatography system on the left-hand side of the screen, and then you've got the automation on the right-hand side. And this is one of our 4-rack systems, which I'll come back to shortly. So just starting it working and just seeing this footage go through. So we start here with the sample probe. It starts in the rinse station. So it's just the probe's been lifted up and it's moved out of the rinse station. This is where the probe gets its internal and external cleaning. And then the probe moves on to the first sample tube in the rack. So what we've got here is 2 50 ml sample tubes in a rack on the deck of the automation. So the probe moves to the sample position. Let's get that running, and it drops down into the position. And the next thing that happens is that the piston pump on the chromatography instrument pulls the sample up into itself, up towards the onto a sample loop, into a column, and it does that via the sample probe here. So what we can see here is that the sample's being pulled up from the sample tube through the probe. Now this is an accelerated process, and what's happening next is that when that whole sample's been pulled up onto the system, it will then be switched into the flow that goes through the column for separation, and we can watch this happening on the operating software of the instrument. In this case, what we're looking at here is Unicorn. And then whilst that separation is taking place, the sample probe returns to the rinse station. So what needs to happen before we move on to the next sample for separation is that this sample probe needs to be cleaned, and we need to be sure that it's not contaminated with any of the previous sample before we move on. So we need internal and external cleaning. So the first thing that happens is we get external cleaning, because in the sample when the sample probe goes into the rinse station, it's being rinsed with clean rinse agent that's being delivered into that rinse station from a peristaltic pump on the rear of the automation. So that's being continually filled and drained, and the exterior of the sample probe is being cleaned. We can also clean the interior of it by rinse being pulled up through the system by action of the piston pump as well on the chromatography instrument. So we get internal and external cleaning of this probe between each sample. And there's plenty of time to do that because the separation takes some time. So we can really take our time cleaning that probe before it moves on to the next sample. And then we move on to the next one. So there we go, just the footage of, we've moved on to the next sample in the system, and then we return to the rinse station again. And we can do that for as many samples as we like on the deck. So what you can see here is that this is one of our sample racks for the 50 ml tubes that has a capacity of 21 positions. And on this particular automation system, we're looking at the space for 4 of those racks. So we're looking at 84 samples that we can run through before we need to refresh these. So that's just some footage of operation for you here. That's some footage that I took of actual samples being run. Now, I mentioned that there's some different types of our automation here. Essentially, we're looking at 2 systems here, and they're essentially the same kind of system. They're based on the same platform. So this is our ASX 500 series platform. And we've got a 2-rack version. So this is the ASX 280 in the middle. And then we've got the 4-rack version, which is the ASX 560. And we've got different configurations of this, which I'll get to shortly. We also have enclosures to go with this. So we are talking about a clean environment in the kind of laboratories that we're talking about for this kind of workflow. So an enclosure would be beneficial for this as well. And that can go with, so what we're looking at here is that particular enclosure goes with the 4-rack ASX 560, but there is a version of it for the 280 here. So just some more details on the actual automation. So I mentioned this is the 500 series auto samplers that we manufacture. This is our industry standard that we've been manufacturing for many years now. And it started its life going into the requirement for automation for trace element analysis with ICP-based instruments. So because it's gone into that kind of market, it's been manufactured with a metal-free liquid-facing flow path. So all of anything that touches liquid going through the system for introduction is metal-free. So it's either based on things like formed plastics, or it's based on things like PFA, etc., or carbon fiber. So this is all designed for trace element analysis, so it's clean from a metal point of view. And we know that in terms of free metal ions, etc., there are some metal ion species that can be detrimental to the actual protein samples that you're interested in. So this is beneficial from that point of view in terms of automating this workflow. We've got various sample volume and capacity options, which I'll get into in a moment, in terms of sample tubes and bottles and sample racks, etc. Also, you can't see really in the image on this screen, but on this slide, but there are some standard positions at the rear of the sample racks as well. So we can fit on this particular 4-rack system, we can fit 10. So there are 10 standard positions for 50 ml tubes at the back. So these can be used for things like additional buffers, reagents, etc. I've already mentioned the rinse station as part of the video footage here, but this is a configurable rinse station for the washout, so we can fill and drain it as required, and we can work with various different reagents to do this to meet the requirements of the workflow. The enclosure I've mentioned. Another thing that's worth noting here is that these systems operate, or can operate, at a low temperature. So as some laboratories require, you could be running this system down at like 4 degrees centigrade. So this could operate in something like a lab fridge for certain workflows where low temperature is required, which it might be for these kinds of samples. So I've mentioned different sample volumes and capacities that you can work with here. So preparative purification workflows, as I mentioned earlier, can work with sample volumes in the low milliliter level or all the way up to the liter level as required, whatever is required for the workflow. So what we have here is the automation can meet the requirements of most of that, going from low milliliter level up to the towards the liter level, but not quite getting there. So we have here, if you've got samples of 1 to 2 milliliters or maybe 5 milliliters or so, then we've got the option to work with something like 7 milliliter sample tubes. If you've got more than that, you could have 15. We have some laboratories that we work with that standardize on a 50 ml sample tube. And then if you needed this kind of volume, then you can work with something like 250 ml sample bottles. So as you can see here, just in these images, so the tube here on the left is the 7, then you've got the 15. The 50 ml sample tube is the one with the blue cap, and then this is a 250 ml bottle. And then we've got all of that is supported by different sample racks. So we've got racks that support each of these kinds of tubes, and then we've also got racks that support the bottles as well. So how does this automation work? How does it function with the actual instrumentation? So if you were to work with one of these in your laboratory, how would it work? So essentially there are 2 connections between the automation and the chromatography instrumentation. The first one is the actual sample probe itself, the one which is liquid facing, that touches the sample and the sample goes up through. So that probe that's on the automation, there is a line attached to that probe, and that line connects directly to the instrument. So it will attach to a switching valve on the instrument so that the piston pump on the instrument can pull the sample through the sample probe, through that line, and then the sample is brought on board onto the instrument. So that's your first connection. And that's a very simple one. It's just a screw fit connection. Now, the next one is how does it communicate? So basically, you have a communication cable, which one end connects to the automation itself, and the other end connects to an I/O communication box on the chromatography instrument. So there's that's your line of communication. So what we do is that because the kind of software that operates the instrumentation itself it can be adapted to work with the different workflows and workflow requirements that are needed. We don't actually change too much on that side. So first of all, we provide with the automation a piece of software, just a small piece of software called AScript. That is where you put your analytical batch. So that's where you put your sample list and your sample positions on the racks, etc. All of that goes into there, and then it just sits in the background and waits to be told what to do. And then on the other side of this, in the operating software for the instrument, we just add a couple of commands. So we add at a certain point in the workflow, and as I said, there's various types of workflow for this kind of instrument, but we add at some point, we command from the instrument tells the automation to go to the next sample position, and the next sample position will be in the batch list that's in AScript. So that sample probe will go to the position that's needed. The operating software will then tell the piston pump that's on board the instrument to pull the sample from that position and bring the sample on board onto the instrument. And then later on in the method, there is another command sent to the automation, and it just says, go do the rinse processes and get ready for the next sample. Simple as that. So we just add a couple of things to the operating software. In this case, what you're looking at here is on the right hand side at the top there, that's our AScript software, and then underneath, just as an example, we have the Unicorn software for the Actor instrument, and a couple of extra commands go in there, and then that's it. So 2 connections, and then a very, very simple approach to the actual operating software, and all of your workflow for introduction can be automated. I mentioned before on the video footage that about the rinse station. I just wanted to sort of re-emphasize this. This is a very important aspect of most kinds of analytical technology. So it almost doesn't matter what it is. If you're automating the introduction of a sample to some form of analytical technology, in this case it's chromatography, if you're doing that one sample to another, then every time you send a sample probe to a sample position, we need to guarantee that that sample probe is clean and it's ready to move on, and there is no cross-contamination from the previous sample into the next one. So basically, that's the reason why we have the onboard rinse station that's supported by a peristaltic pump on the back, so it is filled and emptied by that rinse station. The sample probe is cleaned on the outside just by action of going to the rinse station. And then in terms of the actual interior of the line, that's another aspect of what we can add to the operating software and the chromatography system. So clean rinse agent can be pulled through the sample line onto the instrument, then sent to waste. So basically the interior of the line can be cleaned and the exterior as well. So we can essentially configure what we need here. So if you've got certain samples that are more challenging than others, or have certain aspects of those samples where you need a longer washing time or greater flow rates or whatever it is, or a different reagent, then we can configure the whole washout process to meet those requirements. So just a quick summary for you, really. The automation, basically, my argument here is that by bringing this automation into the workflow for the introduction of samples for purification, basically you can make use of either overnight separations, so you can spend the day doing preparation, bringing samples together into the lab, getting everything ready, and then leaving your analytical batch to run overnight for collection of the fraction you're interested in. Or you can just make the most of a 24-hour continuous separation. You can just leave the system running for as long as needs be to run through as many samples as possible. So again, basically you are removing the need for certain labor in this process, so you don't have to have somebody who's overseeing the system. You can just have that person who is most likely highly skilled and trained, can be going on to do other things in the laboratory, or they can be going on to do the report writing or whatever it is that that's required of them. And then we can configure workflows to make the most of what's needed for any particular project. So that might be that you need to focus on certain sample volumes. It might be that you've got many samples of a lower volume, sort of like at the 5 milliliter level, or that you're looking at separating, purifying a larger quantity of your candidate species from a larger volume. So you can configure the front end here in terms of the introduction to what's required for your project. And then, as I mentioned again, we've got the whole rinsing aspect of this to make sure that everything is clean and ready to go from a sample to sample basis. So essentially better use of staff time with respect to the instrumentation. And this is a very simple, straightforward approach to introducing samples to this kind of analytical equipment. So thank you everybody for your time and for your attention here. Please feel free to visit our website at Teledyne Labs. There's more information on there, or you can contact us directly and we can provide you with any information that you need. If you've got any particular questions, please let us know and we can discuss this further. Anyway, thank you for your time. Thank you for your attention.